Hey anatomists! In this video I'm going to show you how to recognize and identify each of the tissues of the body using photos of histological slides that I've compiled over the past 10 years, and I'll give you tips and tricks to identify these as well. And I'll also give you the location and function of each of these tissues, which could be clues for you on your lab practical, but they'll most likely also be lecture questions for this section as well. All right, let's get to it. Okay, so what I will do for you for each of these tissues is I will actually not display the name of the tissue. That way you can try to guess what the name is for each of these tissues before I actually show them. If this is your first time running through the video, that might not make sense. If you haven't gone through these tissues yet, that might not make sense. But once you run through it a second, third time, fourth time, then you'll see how that'll be useful to you. You'll have a chance to guess what it is before I actually tell you what it is, or before it's actually displayed. And you can slow down the video, speed up the video as you need. I will kind of go fast through these tissues, and you can adjust the speed as you wish from your end. Okay, so I want you to always pay attention to count how many nuclei are in the cells, count how many nuclei you could possibly fit in a cell, and notice the shape of the cell, notice the color of the stain. These things you want to pay attention to. So right off the bat, okay, well I see that we got pinkish. It looks like we have 6 sides, 5 sides for each of the, kind of like a soccer ball patch, and it looks like we can fit maybe 10, maybe 15 of these nuclei per cell. So this will come in handy as we go on to our future tissues. This tissue here is called simple squamous epithelium. Simple squamous epithelium. Why simple? Because every single cell is touching the basement membrane. Now this is a bird's eye view. This is the only tissue where we'll actually see this view from. Maybe there might be one more view, but pretty much this is the only tissue where we actually have the view like this. Why? Well, because they're so squashed, they're so squamous, they're so thin that it'd be hard to see it from the cross-sectional view. So we show it from a bird's eye view. Alright, so why is it called simple? Because each of these cells is touching the basement membrane. Where is the basement membrane? Well, it's below these cells, so you can't see it from this angle. What does squamous mean? That they appear to be squashed. From this angle, again, you can't see that, but you will see that in future slides. So they're squamous means squashed, and make sure you write out epithelium. By the way, along with using this video, make sure to write out these words over and over and over and over. Simple squamous epithelium, simple squamous epithelium. Why? Well, because you'll be assessed by writing them out, most likely. So you practice writing them out over and over and over. Now this particular slide is also called mesothelium, meaning that it's between the tissue and not just separate cells floating around. All right. Location and function. Where is this located? Well, simple squamous, this is a single layer, so you got to be careful of friction. You probably want to put these somewhere where there's almost no friction. Turns out these guys are found in the alveoli. They make up the little tiny air sacs of our lungs, as well as capillaries. The smallest, narrowest of all the blood vessels are the capillaries. And so this is where you want easy gas exchange, easy diffusion. Also the glomerulus of the kidneys, of the nephron, which are also capillaries. So lungs, location, capillaries, another location. What is the function? Well, they're very, very thin, so for easy gas diffusion. Oxygen, carbon dioxide. And of course in the glomerulus you also have other things going through there, which you'll get to in Anatomy and Physiology 2. Okay, so that's simple squamous epithelium. Now I have some more slides to show you of simple squamous. These guys as well, so this slide here, this is also simple squamous epithelium. You may not say that this is mesothelium, where here you would say mesothelium. And technically, I mean, this is actually a cheek, a swab from our cheek, so they look like they're all single cells. So this is simple squamous epithelium. All right, here's our next tissue. Notice how many layers. I mean, look at this. Now, this is a cross section now. Look at all the layers. I mean, look, you can see these cells. They're sort of they're parallel to the ground. They're flat. They may not be so flat as they go down like this, but they are very, very, very long and slender. Lots and lots and lots. This means that this must be not simple, but stratified. This is stratified squamous epithelium. Why stratified? Well, because the only cells that are touching the basement membrane are way down here. All the rest of these are on top of each other, on top of each other, on top of each other. Why squamous? Well, because they look squashed. They look like someone came by and drove over them with a truck. Stratified squamous epithelium. Now, location and function. Well, this happens to be an example of a non-keratinized tissue. Non-keratinized. How can we tell? Because we see nuclei all the way up to the top. Non-keratinized. You find these in the mouth, the oral cavity, the esophagus, the anus, the vagina, anywhere where the tissue cannot dry out. However, in the skin, the outer lips, the outer part of your lips, or in all the rest of your skin should obviously, it can get wet, but it should stay relatively dry. That type of tissue is called keratinized epithelium. Keratinized. How can you tell, or what would it look like if it's keratinized? Well, all of this up here, you won't find any nuclei. Dead skin. Dead skin. That's why it doesn't hurt when when your skin sloughs off, because those cells are not living. But these are living, and we can tell that because you see nuclei all the way up through. So this is an example of, although both are stratified squamous epithelium, this particular slide is showing you non-keratinized stratified squamous epithelium. So this could be an example of like the esophagus or the anus or the vagina, for example, where the tissue should not dry out. It should never dry out. Okay, very good. Here is an example of thin skin. This would be keratinized. This is keratinized. You see how these cells here are, they're translucent. I mean, they're clear. I don't see any nuclei. I see nuclei all through here, but this top layer here this would be an example of keratinized. Keratin is a protein. It's what our nails are made out of, skin, our hair. That's all keratin. It's just a protein. This protein are packed into these cells, but they're not living. These are all dead cells. So these can easily slough off. Okay. In another video, I will show you the difference once we get to skin. I'll make a video specifically just for skin, so we'll talk about all these things, all the layers of the skin specifically. So for now, we're just going to skip that. All right, here's an example of stratified squamous. And what do you think? You see nuclei all the way up to the outside. This must be non-keratinized stratified squamous epithelium. All right, very good. Here's another tissue now. Here's another tissue. Now what we're looking at, we're not looking at all these little circles. What we're looking at is this ring right here. This ring right here. They kind of look cubed. They kind of look like they're like little boxes. Also, the nuclei, if you're looking at how many nuclei you can fit in the intercellular fluid, it looks like you can fit maybe 2 or 3 nuclei into a single cell. You got a cell, 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 cell. That's how you can tell the difference. That's one way that you can be able to differentiate this one from simple squamous epithelium. This happens to be simple cuboidal epithelium. Simple because it's a single layer. Cuboidal because these cells are in, each of them are sort of boxy shaped. Where do you find simple cuboidal? Well, the glands, all of our glands are made out of cuboidal. If you see, these are all oriented in a circle. That's because we've taken a cross section of a tube of a gland. Glands are just made out of tubes. And so this is an example. This could be the pituitary, for example, or nephrons, or it could be the pancreas, or even the thyroid. And so what is the function? Well, what do glands do? They generally secrete, but you also, in the nephron of the kidneys, you can also have absorption as well. Now, be careful, because not all simple cuboidal slides will be from this view. You might see, here's another one here. This looks very similar to this one. You can see even the nuclei even more clearly. You could fit maybe 2, 3 in there. However, we could cut the tube down the length, and therefore you won't see circles. You see, this is cutting the tube down the length, cutting the gland down the length. So just pay attention to that. It's not every time that you see every single simple cuboidal tissue slide will be circular like this. Not necessarily. Here we're seeing a section down the length of the tube, but you do see the cuboidal cells here, the shape cubed. Okay, and of course they're simple because there's a single layer here. Okay, very good. And then, so where would the basement membrane be then? So then the basement membrane would be right here. This is the inside. Here's the lumen right here. Here's the inside of your tube, of your gland. Okay, very good. All right. Okay, for this one, it looks like we have a single layer here and then another layer on top. At least 2 layers going around in a circle. This is stratified cuboidal epithelium. Stratified. Where do you find these? Well, these are also glands. This is glandular epithelium, but you'll find these specifically in salivary glands, sweat glands, mammary glands, but the function is the same as with simple cuboidal epithelium. The same function, but you find not all glands are simple cuboidal epithelium. There's a few that are stratified like this. The locations of these guys would be your salivary glands, that would be in your mouth, your sweat glands, in your skin, as well as the mammary glands. Okay, very good. All right, what we're looking at here is this is one cell. Check this out. I can start by looking at these nuclei. Very neat and organized. Well, if I were to outline the plasma membrane, it would go like this. A very, very, very long, slender box, almost like a column. The basement membrane is over here. And it looks like, if you notice, there's like this dark layer here. These are microvilli. These are tiny little hairs called microvilli. The nuclei are here, and the plasma membrane goes around like that. This is one single layer of cells, but they're column-like. They're very long. This is simple columnar epithelium. It starts from here and goes all the way to here. You can also see them on this side. The nuclei are very organized. They are right next to each other, and that's how you could tell this is simple columnar epithelium. Notice the goblet cells. Goblet cells secrete mucus. And then you see the line of microvilli. Tiny, you have to zoom in even more, but you got these little hairs going like this. The longer hairs are called cilia, and the shorter hairs are called microvilli. Where do we find these? Well, in the stomach, The small intestine, large intestine, that sounds like most of the GI tract. So, like the GI tract. You also have them in fallopian tubes. You'll find simple columnar in the respiratory bronchioles, which are the first area where you have the first place where you have respiration. And function, absorption and secretion. Absorption and secretion. Simple columnar. Let's take a look. Here's another view. Another slide. Look, here's a goblet cell, goblet cell, goblet cell. And the nuclei, it looks like, we're talking very neat and organized, but they're very long. Columnar, they're column-like. Here's another example, another slide, from here to here. This one might be a little bit tougher to see that, but again, these are the nuclei going all the way around. And the nuclei, they do tend to sit closer to the basement membrane. You see, they are a little bit closer to the membrane, kind of like on the bottom, versus the apical. The top or the apical surface, that would be like here, and down here, this is the base closer to the which is touching the basement membrane. Simple columnar epithelium. Here's another example, simple columnar epithelium. All right. Looks like we got columns here. I do see columns, but then these guys, they look like they're almost going the other direction. Well, these guys are rare. Universities and colleges, they may not even have this slide. And that's because the tissue is so rare. This happens to be stratified. There's two layers here. Stratified columnar epithelium. Stratified. Now these guys don't look like column-like to me. Well, yeah, they're not column-like, but overall, these guys are, so we just call the whole thing stratified columnar. So this is stratified. Where do you find these? In the conjunctiva of the eye, the parts of the pharynx, not all the parts of the pharynx, but parts of the pharynx, the uterus, and the same function as simple columnar epithelium, which is absorption and secretion. So this is stratified columnar epithelium. All right, I see hairs, but they're longer. They're much, much longer. These are cilia. These are the longer hairs. These aren't microvilli, but these are cilia. The nuclei, it seems to be a big mess. It's just a big mess. It looks like there's some here, 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 here. It almost looks like it's stratified. It turns out this is not stratified, but it looks to be stratified, so we call it pseudo-stratified ciliated columnar epithelium. Pseudo means false, stratified means multiple layers, so it's falsely stratified. Make sure to have this in the correct order as well. Pseudostratified ciliated columnar epithelium. Where do you find them? This is the upper respiratory tract, as well as some of the lower in the trachea. You'll find goblet cells, not in this particular side, but you'll find goblet cells. Now there is such a thing as non-ciliated pseudostratified columnar, like in the prostate gland and in the membranous parts of the vas deferens, but for the most part, it is ciliated. The pseudostratified is ciliated, and these guys secrete. So the goblet cells will make mucus. They'll send it up to the top here, trap pathogens, and these cilia will actually move the mucus all the way up and out the trachea, all the way to your mouth, and this mucus traps pathogens, little ickies, and then all you got to do as it comes up, you don't feel it, of course, you don't feel the cilia moving, it just happens, and then what happens when you have a lot of mucus, or when you get enough mucus, what do? You swallow, it goes down the esophagus, down to the acidic stomach, and kills any pathogens that have been trapped in the mucus. It's called a mucus escalator. Mucus escalator. So that's your pseudostratified ciliated columnar epithelium. You zoom in, like, wow, okay, this does look stratified. Look, you got a nucleus, nucleus, nucleus. It looks stratified, but it's not, so we call it pseudostratified. Look at how long the hairs are here, the cilia. Here's a goblet cell. There's a goblet cell. This will also give it away. So how can I tell this from, say, simple columnar? Well, the nuclei are on top of each other. And you also have really long cilia. That's pseudostratified ciliated columnar epithelium. Here's another example here. Here's another slide. Pseudostratified. All right. Let's see here. I don't see any cilia in this one. It looks like you got these oval-like cells, but they're perpendicular to the ground. Perpendicular. They're standing upright. But then you have these ones that are going horizontally. This is transitional epithelium. Transitional. The location of these is the urinary bladder, the ureters, and the urethra. And what is the function of this? Well, the urinary bladder, as it fills with urine, it has to take up all that urine, and so it has to be able to stretch. So it's very stretchy, but it goes back and forth. So it transitions, and when it stretches, it looks different. And when it goes back, when you empty when you void, it'll look like this. So it's called transitional because the tissue does actually transition in shape, because it has to be able to take the shape of whatever is going through it, whether it's in the ureters and the urethra or in the urinary bladder. Transitional. This is the only one that looks like this. Here's another, all of this here. Here's transitional epithelium. It kind of looks like a big mess, and it's the one that's really kind of hard to even describe, really. The cells are kind of going vertically for the most part. There's a lot of them on top of each other. That's all transitional epithelium. Okay, hopefully your professor will sort of cut you a break on this one. Because it does look like a few others. This is smooth muscle. Smooth because it doesn't have the zebra print going perpendicular. It doesn't have the striations. Now, you notice that there is a lot of white between. There's a lot of white, at least in this slide, between each of these. And it looks like we got nuclei. This is smooth muscle. Smooth muscle. Where is smooth muscle found? Smooth muscle is found in the artery walls, in the GI tract, anywhere where you need muscle to move food or air. That's not skeletal muscle. That's not attached to your bones. If muscle is attached to your bones, it's called skeletal muscle. Pretty much all other muscle is called smooth muscle. This is all involuntary. Of course, you have your cardiac muscle. That's the third kind of muscle. All right, so this is smooth. This is smooth. Sometimes the slide is really, really, really light. That's because it's the way it is. That's the stain. But you do see a lot of white in between. Here's another example of smooth muscle. It's white, 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 white. It looks like the muscle cells, the fibers themselves, are very slender. Here's an artery. Smooth muscles going around in a circle. Here's an artery. Okay, now this is also muscle. However, we do see this zebra print, black, light, dark, light, dark, light, dark, going perpendicular to the length of the fiber. This zebra print, these are called striations. Striations. This is skeletal muscle. Skeletal muscle. You find skeletal muscle attached to your bones. That's why attached to your skeleton. That's why it's called skeletal muscle. Now, It is hard to find really good stains like this, so sometimes you'll need a clue. But hopefully, this was a very, very hard one to find. Sometimes you can't. Sometimes skeletal muscle, it'll look like cardiac or like smooth muscle. But obviously, you got the striations going perpendicular. These little etch marks going perpendicular to the fiber, and this could be a lab practical question, which is, what are these marks going this way? And the answer is striations. So these are striations. Here's some more. You see them going like this. Okay, now, the difference between these and this next one is that another difference is that these fibers are very parallel to each other. They're flat and they're parallel. They're flat and they're parallel. You see that? But this one, it looks like these fibers are branched. They're sort of branched back and forth, back and forth. That's because they are. That's because they're all attached to each other. This is cardiac muscle. Cardiac muscle is generally stained purpley like that. You'll also find every once in a while, these ones are not striations. These are called intercalated discs. An intercalated disc marks the beginning of one muscle fiber and the end of another. Intercalated discs. Now, cardiac muscle is actually striated, but it's so faint that it's hard to see. If you were to zoom in, you will eventually be able to see the striations. So they are striated, but don't let that throw you off. This is cardiac muscle, and where you have branched fibers and the intercalated discs are running perpendicular. But these ones, so this one here, this one here, this one here, this one here, these are not the striation. These are the intercalated discs. All right. This kind of looks like smooth muscle, doesn't it? It kind of looks like smooth muscle, but there's a lot less white. These bands, these pink bands, are a lot wider than with smooth muscle. Let's take a look at smooth muscle again. Like, oh yeah, these are much thinner, much thinner. Whereas with these, they're much wider. These pink bands are much wider. This is collagen inside. This is all collagen. The broad pink bands is all collagen. What type of protein fiber is this? This is collagen. This tissue is dense regular. Dense regular connective tissue. Make sure to write it all out. Dense regular connective tissue. Why is it called dense? Because there's two types of connective of CT proper. There's dense and there's loose CT and dense CT. Why is it called regular? Because these fibers are fairly parallel with each other, packed. They stack very nicely, so that's called regular. Regular. Where do you find dense regular CT? We find this in our ligaments and tendons. They take tension very, very well from two directions, but not very well from the other two directions. So from these two ends, they take tension very well. They can take stress very well, but they tend to tear very easily if there's tension applied this direction, which is why we always tear our ACL and that kind of thing. Here's another example of dense regular CT with the collagen inside. I mean, these are collagen fibers. And here, although you see waves, they're still parallel with each other, so it's still dense, regular CT. All right, here's another type of tissue, and it looks, we got like wavy, it looks like, I don't know, we got wavy lines. Well, this is elastic tissue, and these wavy protein fibers are elastic fibers. So you have elastic tissue made up of elastic fibers. Here's another example, elastic tissue. Elastic tissue. These could be surrounding arteries, because arteries are very elastic. They have to be able to take absorb the blood as it goes through and shrink once again, vasoconstrict, vasodilate. Now, One more thing that I wanted to mention is that there's actually three elastic terms. There's elastic tissue, there's elastic fiber, but there's also elastic cartilage. So just to keep you from being confused, to help with that, there are three different type of elastic answers for three different questions: elastic tissue, elastic fibers, and then we'll get to elastic cartilage. Okay, this is, I already showed it. This is dense irregular CT. Dense irregular. Dense because it belongs to the dense proper, but also irregular because this collagen seems to be going every which way. Look at this. You got fibers. They're not regular. They're not parallel with each other. You got fibers going into the screen, out of the screen, every which direction. It's still made out of collagen, that pink stuff, but because it's a big mess, fibers are going every direction. It's called irregular, dense irregular connective tissue. Now, what is the advantage of dense irregular? Well, because the fibers are going in every direction, this tissue can take tension from all kinds of directions. This is actually found in the dermis of the skin, more specifically in the reticular layer of the skin, dense irregular CT. And, well, yeah, take your cheek and just pull in any direction. Take your cheek, take your arm, pull your skin, take your leg skin, pull it any direction, and it will pull and it'll go right back. It can take tension from so many, whereas if you take a tendon or a ligament, it can't stretch in just any direction. It won't, actually it doesn't stretch at all, but I mean it doesn't take tension. That's the advantage of dense irregular, is that although it's not as strong in any one direction, it does take tension from many directions. That's dense irregular CT. Here's another example, dense irregular CT. All right, this one in my opinion is the coolest. It looks like somebody took like a box, a cardboard box, they put some marbles in it, they put some different color paints in there, and they sort of they shook the cardboard box around and let the marbles float around. That's what that looks like to me. You got all kinds of different fibers. We have that pink broad in the background. That's once again collagen. That's collagen. But then you also have these elastic fibers. These hairs, these hair-like fibers. That's elastic. This is areolar connective tissue. The whole word would be areolar loose connective tissue. Areolar connective tissue. What's at least one place that we could find areolar? This is also in the dermis, but this is in the upper layer of the dermis. The dermis can be broken up into two layers, two sublayers. The papillary layer and the reticular layer. Well, the papillary layer, the top 1/5, is made up of a tissue called areolar connective tissue. And it's made out of all kinds of cells and fibers, like elastic fibers and collagen fibers. Here's another example. Like, it's very distinct, you guys. Like, there's no tissue that even comes close to looking like this one. Like, this is right off the bat. You should just know right off the bat, this is areolar CT. Okay, this one is pretty much clear. I mean, this is clear. There's almost nothing here. That's because this is adipose connective tissue, adipose CT. Very clear. Now, do you remember all the way back to our first term, our first slide? That was simple squamous epithelium. This does, in a way, look like simple squamous. You got those multiple sides. However, the nuclei, they're gone. Where are they? Well, they're tucked in the corners. This is fat. This is fat tissue. Adipose is fat. How I say it is, the fat cells are so fat that they push the nuclei into the corners. That's how I remember it. So the nuclei are there, but they're in the corners, not somewhere in the middle like you would with simple squamous. I say that simple squamous epithelium looks like a sunny side up egg, like a fried egg, whereas with adipose it doesn't look like that at all because you don't see the nucleus in the middle there. So this is adipose CT. Where do you find adipose? Well, you find it below the skin. The skin has two main layers, epidermis and dermis, and the hypodermis, or below the skin, is primarily made out of adipose CT. All right. Here's another. Here you can see the nuclei, but again they're squashed in a corner. They're kind of squashed in a corner. So it's very obvious that this is not simple squamous. One cell is called an adipocyte. Adipocyte. Anytime you have the word cyte at the end, that means cell. Cyte means cell. Adipocyte. Here's another one. Very clear. That's because fat is pretty much white. I mean, it's like a yellow. White is yellow. So in a slide, it's very very translucent. All right. Here is reticular tissue. Now, these little black branches, it almost looks like little twigs, little sticks. I call them Olaf arms. These guys are reticular fibers. So now we've been introduced to the three fibers that we're going to need to know. Collagen fibers, those were the broad pink. Elastic fibers, those were the squiggly or the hair-like looking ones. And then we have reticular fibers, which are these kind of branched, sort of dark structures, like twigs that you'd find on the ground, like Olaf arms. Those are the reticular fibers. And you have white blood cells. Where do you find reticular tissue? You can find reticular tissue in the spleen, lymph nodes, red bone marrow. That's where you would find this stuff. These are all lymphocytes here. Okay, so that's reticular tissue. Here's another slide of it here, reticular CT. Okay, now it looks like we're looking at this centerpiece here. Looks like we got a nucleus. It looks like we have a pink dot, and then white around it. And then we have this. The cells are definitely not touching each other. They're spaced out. It turns out that this is hyaline cartilage. This is hyaline cartilage. Now, the nuclei that you see here are not nuclei. These are not the nuclei. The pink dot that you see in each of these white things, those are actually the cells. And the white that you see around is the lacuna. Lacunae. So one is lacuna. What is the lacuna? It's the home. It's the home of the cell. What is the name of this cell? Well, this is cartilage, so this is a chondrocyte. Chondrocyte inside of the white little home called lacuna. Lacuna. This cartilage here is called hyaline cartilage. What's the location? Where do you find hyaline cartilage? You find hyaline cartilage at the ends of your long bones. You also find them attached to your ribs. What else? The trachea. Some of the trachea. This is all hyaline cartilage. All these things are made out of hyaline cartilage. Very rigid. Very rigid. It's not elasticy at all. Very rigid, like at the ends of your long bones. How I remember it, hyaline cartilage, you just wave your hand high, hyaline. So your arms are made out of long bones. And at the ends of your long bones, they're wrapped with this articular cartilage called hyaline cartilage. All this is hyaline. And the matrix is very well, let's say this is like a paint. Let's say you mix this up. You don't see any streaks in here. It's very well mixed like a watercolor. Whereas the next tissue that I'm going to show you, this is fibrocartilage. And in the matrix, you do see like streaks. You do see streaks in here. Also, the lacuna are more slender-like. You see, they almost look like eyeballs. They're more slender. Well, eyeballs in the eye socket, so they look like eyes, like that you would draw. Oval shape, kind of. Whereas with the hyaline the hyaline cartilage cells and the lacuna and their lacuna, they're more plump and round. You see that? Here, this is fibrocartilage. This is not the very rigid and not the very elasticy type of cartilage. This is the stuff in the middle, like in your vertebrae, your intervertebral discs. Fibrocartilage, again, you see the streaks in the matrix. Streaks in the matrix. And then finally, this one is very, very, very obvious, very distinct from the other two. This is your third and last type of cartilage. You see the lacuna. Look at how, I mean, these cells are like almost touching each other. Whereas these, they're very spaced out, very spaced out, very spaced out. But this cartilage, they're like, there's like no room for the matrix. This is elastic cartilage. Elastic cartilage. Here's another example. Elastic cartilage. The lacuna are just ginormous. They're just huge. Where do you find elastic cartilage? You find elastic cartilage in the pinna, the external ear, called the pinna. It's very elasticy. It's not rigid at all. It's the least rigid of the three types of cartilage. In the epiglottis, also, you'll find elastic cartilage. Here's another example. All these are elastic cartilage. Elastic cartilage. All right, this is compact bone. Compact bone. Make sure to say the whole word, compact bone. I mean, the whole thing really is compact bone connective tissue, but people don't really talk like that. Compact bone. And then we got different. We have. Once we get to the bone chapter, we'll do this even in more detail. But here's a few things that you'll want to know. You see this unit? You have like a whole unit here. There's another unit here, another unit here. This whole thing is called an osteon. Osteon, with a central canal in the middle. Central canal, and you have these layers called lamella. These are concentric, going around in a circle, concentric lamella, and then you have these little etch marks. You see these etch marks here, going perpendicular to the layer. These are little canals. These are canaliculi. Canaliculi. These are little canals. So you got you have the whole thing called an osteon. You got the central canal. You got the canaliculi, these little etch marks, dark etch marks going perpendicular here. Concentric lamella going this way. And then you have the osteocyte, which are the black dots. The osteocyte is they are technically in lacuna, just like with cartilage. They are in lacuna. It's hard to see. So if you're asked what is this black dot, then you would say osteocyte. This is the mature bone cell. If the question is what is the space that the bone cell is sitting in, then you would say lacuna. Lacuna. Here's one layer. See, here's one layer. Here's a concentric lamella. Okay, very good, you guys. Compact bone again. All right, here's another tissue called blood. Fluid tissue. This is also connective, a type of connective tissue. This is blood. What are the components? Well, you got these red blood cells here. Red, all these, most of these dots here, these are all red blood cells. And then you have these larger cells here. Here's one, two, I see two larger cells. These are white blood cells. You have the watery part here, all this white space. This is the plasma. And then you have the little cookie crumbs. The little cookie crumbs, these are your platelets. Platelet. These are your little cookie crumbs. Okay, so I don't have it here. Red blood cell. So I guess I don't have it written here. Red blood cell, white blood cell, platelets and plasma for your blood. Okay, and this is nervous tissue. Here's the body of a neuron. Here's the body of a neuron. How would you describe this? Well, a lot of times the stain is orange, but not always. Sometimes you do see this intricate network. You do see a lot of times you do see the bodies having like these extensions. So you see how this one is orange, but this one doesn't have that orange stain. So this is nervous tissue. Glial cell. So you'll have, these large ones here are neurons. These are the cell bodies. The nucleus, the cell body of the neuron, and then you'll have smaller ones called neuroglia, or glial cells, the supporting cells. Here's another slide here. This is nervous tissue. I can't really think of a tissue that looks like this one, so, you shouldn't get this mixed up with any other tissue. Okay, so that concludes the tissues, the histology of all of the tissues of the body. You'll want to run through this over and over again and practice writing them down, writing out the words over and over and over. And the words are, these terms really are, they're intimidating. I'll be honest, they're intimidating. We've never heard of these words. It's also intimidating because everything here is microscopic. It's not things that we can touch and hold like bones and muscles. That's just the way it is. We start with this chapter in your whole year of anatomy because we take a little glance of all the tissues, and then we start to go through all the systems one by one. All right, thank you guys very much. You'll find another video for skin, and we'll go through thick skin, thin skin, keratinized, non-keratinized, all the parts for tissues and for the skin models. All right, thank you guys very much, and study well.